Hello students, welcome back to the biology class. So we carried on with the topic of biotechnology principles and processes in the last class, and we learned about the first instance of construction of the artificial recombinant DNA. As per the NCERT, this was done by Stanley Cohen and Herbert Boyer. We learned that in 1972 these two scientists successfully linked a gene encoding antibiotic resistance first of all with the plasmid of Salmonella typhimurium as per NCERT. Otherwise there is difference in the books of the biology of references type. And then from the Salmonella typhimurium they successfully cut it out with the help of molecular scissors and transferred this gene which provides resistance against antibiotics into the E. coli. And we learned that it successfully became a part of the plasmid of E. coli, and thus this piece of DNA transferred into E. coli, which is closely related to Salmonella, was able to replicate using the new host's DNA polymerase enzyme, and thus it multiplied to make copies there. And this ability of multiplying the copies of antibiotic resistance gene in the E. coli is called as cloning of the antibiotic resistance gene in E. coli. We learned regarding the tools of the recombinant DNA technology in the last class. We learned that there are enzymes which are called as restriction endonuclease enzymes, and we understood the background regarding the nuclease enzyme in the last class, that restriction enzymes belong to a class of the enzymes which is called as nuclease. It's a large class, and this class includes two types of enzymes, exonuclease and endonuclease. So as per the NCERT, exonuclease removes nucleotide from the ends of the DNA, whereas endonuclease makes cut at specific positions within the DNA. So we learned that each restriction endonuclease functions by inspecting. So how does it function? First of all, it functions by inspecting the full length of DNA. So longer the length of DNA, more time the enzyme is going to take to cut more pieces of the DNA. If less time is given, then also you will find less number of pieces will be there. Once it finds its specific recognition sequence, we learned the example of Malayalam, that is a palindromic word, that the words which read same in the forward direction and backward direction are called as palindromic sequence. Similar sequences are found in the genetic material, and they are called as the palindromic nucleotide sequence. So once it finds a specific recognition sequence, we learned that in the last class that it binds to the DNA, then cuts the two strands of the double-stranded helix at the specific points, and it always makes cut in the sugar phosphate backbone. We understood the topic of the sticky ends also in the last class. We understood that the enzymes required are the lysing enzymes. Lysing enzymes are those enzymes which are supposed to be opening the cell so that DNA can be taken out. And the first enzymes required are those which break down the cell wall. So if it is a bacterial cell, then we need lysozyme. If it is a plant cell wall, we need a cellulase. And if it is a fungal cell, example rhizopus, then we need the chitinase enzyme. After breaking the cell wall, we learned in the last class that we need different other enzymes to break down proteins, protease, to break down lipids, lipase, and to break down the RNA, RNase enzyme is required. Once the DNA has been extracted by this process, then we need the cleaving enzymes. Now cleaving enzymes are those which breaks down the DNA molecule, and we learned that exonuclease always make cut at the terminals, and it always make cut in the single strand of the double helix, and it does not cut the RNA. We learned that endonuclease always cut at both the strands. It can cut at the single strand also, but it can even cut the RNA, but it reads a particular sequence to cut. And restriction endonuclease are those which read a particular sequence on both strands. It is going to make cut at a particular position only, and thus it may lead to the formation of either sticky ends or otherwise blunt ends. We learned that there are three types of restriction endonuclease, and type 2 is the one which we always use for the biotechnology or in the molecular engineering concept. Remember, type 2 is of two type. Again, some of the enzymes create sticky ends, some of them create the blunt ends. In today's class, even the blunt end will be clear to you. So this much we covered in the last class, and I told you the definition of sticky ends, that we learned the example of XII in the last class, reading from 5' to 3', that when the E-core enzyme, is made to cut at both the strands, it will read a particular sequence from 5' to 3' direction and makes a cut always away from the center. This is the center, away from the center. So it is always between G and A. It is always between G and A, away from the center. So once a cut is made, now there will be 4 bases which are free and they are single stranded. So this single strand overhangs are called as the sticky ends, and they are called sticky ends because they are able to form hydrogen bonds with their complementary cut counterparts, and this is done with the help of the DNA ligase enzyme. So we covered this much in the last class.